Hey all, welcome to my channel, and today I'll be discussing regarding metabolism. So, speaking about what metabolism is. Metabolism is a series of interconnected reactions that occurs within a cell. Metabolism is very crucial for the normal growth, development and functioning of our body. Okay, so metabolism could be categorized into two important types, that is one being the anabolism and the other is catabolism. We'll discuss each in detail, quoting examples, right? So, large macromolecular molecule is built up using smaller, simpler units. For example, amino acids are used for the building up of complex polypeptide or protein. Okay, in anabolism there is a requirement of input of energy, right? Catabolism, on the contrary, it refers to breakdown of larger complex molecule into smaller, simpler raw materials. That is, lipid is broken down to fatty acids and glycerol. In catabolic pathway or in catabolism, there is a release of energy which the body can make use for its later use. Okay. Now, the fashion in which metabolic pathway runs categorizes it into three important types. First one being linear pathway, which we'll be discussing today, under which we'll be studying glycolysis. The second one being cyclic pathway, cyclic because it runs in a cyclic fashion. One example is citric acid cycle, and the third one being spiral pathway, and an example of it is biosynthesis of fatty acids. Righto. Now, glucose. We are very much concentrating on glucose because in the subsequent slides we'll be discussing how glucose is broken down and is made use to release energy, right? So where do we find the use of glucose in a body? Glucose is a prerequisite for the synthesis of structural polymers, that is for building up of ECM, extracellular matrix, and cell wall polysaccharide. Glucose is a must, right? And via oxidation, glucose is oxidized via the pentose phosphate pathway to give rise to ribose 5-phosphate, which is an important component of DNA. Also, glucose is stored in different forms. In plants it's stored as starch, in animals it is stored in the form of sucrose, glycogen, etc. Right? Also, glucose, which we'll be now discussing, undergoes oxidation reaction via the glycolytic pathway to give 2 3-carbon molecules pyruvate. Okay, so talking about the glycolytic pathway, essentially all cells, they carry out glycolysis. So glycolysis is basically a 10 enzyme catalyzed reaction, right? The 10-step enzyme catalyzed reaction is categorized into two phases. The first one being the preparatory phase and the other one being the payoff phase. The first 5 steps of glycolysis are referred to as what we call as preparatory phase of glycolytic pathway. So we'll have a just flow chart here. We'll just go through the flow chart, and later in the subsequent slides I'll be teaching each step in details. Righto. Now here in the preparatory phase, the first 5 steps include conversion of glucose to glyceraldehyde 3-phosphate and DHAP. That is the 5th step of preparatory and final step of preparatory phase. Here glucose is converted to glucose 6-phosphate with the help of enzyme hexokinase. Okay. In the second step, glucose 6-phosphate is isomerized to fructose 6-phosphate, and third step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. In the fourth step, fructose 1,6-bisphosphate is split into 2 3-carbon molecules. One is glyceraldehyde 3-phosphate and the other is DHAP, that is dihydroxyacetone phosphate. We'll discuss each step with enzyme being used and the cofactors, right? Next, this is the payoff phase of glycolysis. So in the payoff phase begins with glyceraldehyde 3-phosphate. Here actually what happens is there will be availability of 2 glyceraldehyde 3-phosphate, as DHAP could be converted to glyceraldehyde phosphate again, right? So glyceraldehyde 3-phosphate is converted, 2 molecules of glyceraldehyde 3-phosphate is converted to 2 molecules of 1,3-bisphosphoglycerate. Okay, 1,3-bisphosphoglycerate converted to 3-phosphoglycerate, and 3-phosphoglycerate is converted to 2-phosphoglycerate. 2-phosphoglycerate is converted to phosphoenol pyruvate, and finally phosphoenol pyruvate is converted to pyruvate. This is the glimpse of glycolytic pathway. So glycolytic pathway has two phases. One is preparatory phase and the payoff phase. The first 5 steps we refer to as preparatory phase, and the subsequent 5 steps is referred to as payoff phase of glycolysis, right? So here this is also, we'll discuss this slide later. And now talking about the first step of glycolysis. What happens in the first step? In the first step of glycolysis, 6 carbon molecule glucose is converted to glucose 6 phosphate, meaning to say it is phosphorylated with the help of enzyme hexokinase. One thing keep in mind, whenever you hear about the enzyme kinase, it's very important that for the activity of kinase, magnesium ion and ATP is a prerequisite, without which the reaction, it cannot carry out the reaction. So whenever we hear about the word kinase, what will strike to our mind? Magnesium ion and ATP. So as I said here, glucose is phosphorylated. Where is it phosphorylated? Glucose is phosphorylated at the 6th position, 6th carbon. A phosphate group is added. So I said ATP is being used. What's the use of ATP? So the phosphate group that is attached at the 6th carbon of glucose is borrowed from ATP, itself reducing to ADP, right? Adenosine triphosphate, there were 3 phosphate initially, has donated one of its phosphate to glucose, and it is attached at its 6th carbon, and itself it's reduced to diphosphate now. Okay, and what is the enzyme that catalyzes this reaction? That is hexokinase. Glucose phosphorylated to glucose 6 phosphate with the help of enzyme hexokinase. So this is the first and committed step of glycolysis, and it's an irreversible step. It's very important. Keep that in mind, right? One fascinating thing here is the importance of phosphorylation of glucose is, once glucose enters into a cell, there are chances that it might escape the cell, right, via the plasma membrane. So what holds on glucose inside the cell? Yeah, it's the phosphorylation. Once glucose is phosphorylated, it has no way to escape the plasma membrane, and therefore it retains inside the cell. That is the importance of hexokinase and phosphorylation of glucose, right? So the phosphorylated glucose is isomerization reaction occurs, that is it's isomerized to fructose 6 phosphate. So what's the difference here? Nothing. There is not much difference. What happens here is aldose is converted to ketose. Aldoglucose is converted to ketofructose, right? Glucose 6 phosphate is isomerized to fructose 6 phosphate with the help of enzyme phosphofructoisomerase. Also we can call it as phosphohexose isomerase, right? This is the second step, and it's a reversible step of glycolysis. Next, fructose 6 phosphate is converted to fructose 1,6-bisphosphate. Now what happens? Now ketose to ketose. There is no conversion of aldo-keto, nothing like that. Okay, fructose 6 phosphate, already a phosphate group is present at the 6th carbon position. Now one more phosphoryl group is added at its 1st carbon position. Again here the enzyme used is phosphofructokinase 1. Why 1? PFK1, because PFK2, the same isoform, is used elsewhere, right? We'll be discussing it later in the subsequent lectures. So phosphofructokinase converts fructose 6 phosphate to fructose 1,6 diphosphate or bisphosphate. Here the phosphoryl group is borrowed from ATP, itself getting reduced to ADP, and the so donated phosphate group is attached at fructose carbon 1, right? And this is again an irreversible step of glycolytic pathway, and it's very important committed step of glycolysis, right? The first step is irreversible, second is reversible, again the third step is irreversible step. Next, this is quite fascinating now. Fructose 1,6-bisphosphate so formed is acted upon by enzyme aldolase. What happens here is aldolase enzyme has got the capacity to break down 6 carbon fructose into 2 3 carbon molecules. One is dihydroxyacetone phosphate and the other is glyceraldehyde 3-phosphate. So what happens in the fourth step? With the help of enzyme aldolase, fructose 1,6-bisphosphate is broken down to two 3-carbon units, DHAP, and DHAP so formed is again isomerized to glyceraldehyde 3-phosphate. So what can we say is, from one glucose molecule, two glyceraldehyde 3-phosphate molecules could be generated in each glycolytic pathway. One is directly from fructose breakdown, fructose 1,6-bisphosphate breakdown, and the other is the subsequent isomerization of DHAP back to glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate so formed is converted to 1,3-bisphosphoglycerate with the aid of enzyme glyceraldehyde 3-phosphate dehydrogenase. So what happens here? In this step, glyceraldehyde 3-phosphate makes use of the inorganic phosphate present inside the cell, and it subsequently reduces NAD plus to NADH and proton with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase, giving rise to 1,3-bisphosphoglycerate. As the name suggests here, the phosphoryl group so taken up from the cell is added at carbon 1. Therefore, two phosphoryl groups are added. One is at carbon 1 and the other is at carbon 2. Therefore, giving rise to 1,3-bisphosphoglycerate. Glyceraldehyde 3-phosphate taking up inorganic phosphate and reducing NAD with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase, it is converted to 1,3-bisphosphoglycerate. Now, the next is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. And this is one of the most important, and we can call it as a committed step of glycolysis, because here there is generation of first ATP. What happens here? 1,3-bisphosphoglycerate donates its phosphoryl group to ADP, converting ADP to ATP, and itself getting converted to 3-phosphoglycerate. So with the help of enzyme phosphoglycerate kinase. Phosphoglycerate kinase, as I said, it requires the cofactor magnesium for its activity. It converts 1,3-bisphosphoglycerate to 3-phosphoglycerate. The phosphoryl group present at carbon 1 is now donated to ADP so that it forms ATP. Since here in this step there is ATP generation, this step of glycolysis, that is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, is referred to as the first substrate level phosphorylation step. Why is it called substrate level phosphorylation? Because the substrate of glycolytic pathway, one of the substrate, is helping for the generation of ATP, right? The 3-phosphoglycerate so formed is converted to 2-phosphoglycerate. There is just a shift of phosphoryl group from carbon 3 to position 2. This is accomplished with the help of enzyme phosphoglycerate mutase. So this enzyme shifts the phosphoryl group from carbon 3 to carbon 2. The 2-phosphoglycerate so formed is converted to phosphoenol pyruvate with the help of enzyme enolase. Always keep in mind, whenever you hear about enzyme enolase, it is the enzyme which brings out water molecule. Okay, the enzyme which brings out water molecule is enolase. Enolase takes out water molecule from 2-phosphoglycerate, resulting in the formation of phosphoenol pyruvate. In the pink shaded area, you can see H2O is being released, and the remaining atom structure what we get is phosphoenol pyruvate. The phosphoenol pyruvate so formed is finally converted to the final end product of glycolysis, that is pyruvate. What happens here? Phosphoenol pyruvate donates its pyruvate phosphoryl group to ADP, again generating ATP with the help of enzyme pyruvate kinase. Pyruvate kinase converts phosphoenol pyruvate to pyruvate by donating the phosphoryl group from PP to ADP generating ATP. So this is the last and final substrate level phosphorylation step of glycolysis. In total, there are two substrate level phosphorylation steps that occurs in glycolysis. One is while converting 1,3-bisphosphoglycerate to 3-phosphoglycerate, and the other is last step, that is conversion of PEP to pyruvate, right? And this completes glycolysis. So in total, one glucose molecule is converted to two pyruvate molecules, right?